Thank you for Lauren for a nice introduction. It's really my great pleasure to take this opportunity to discuss another non-coding small RNA. So called here. So I focus on enhancer RNA. As you know, enhancer are key class of non-coding regulatory elements. So in the genome, one linear space, that enhancer may be far from the target gene, but in the 3D structure, the enhancer would close to the promoter region, facilitate the recruitment of transcription factor and turn on the gene. So this is really an important small non-coding region. So previously, people focus on enhancer RNA usually quantify their activity based on ChIP-seq, but that's actually not very convenient, especially for large-scale clinical sample. One alternative way to quantify enhancer activity is to quantify the enhancer RNA, or we call them eRNA signal. So from this figure, you can see for inactive enhancer, those enhancer region, they are well-ramped among nucleosome. And then in the pulse enhancer, there's only a very narrow nucleosome-free region. But when this enhancer is fully activated, the whole region, you have this wide nucleosome-free region. So they also can recruit transcription factor polymers too, and then they generate that short RNA, often have this poly A. So that short RNA, we call that enhancer RNA. So in this way, you can use that enhancer RNA as a readout for enhancer activity. So but usually, when we want to quantify enhancer, we need to know exactly the location. So enhancer locus can be captured by cage. So here, basically, it's the experimental procedure by the cage. Using this technique, there are some consortiums such as Pantone has quantified the enhancer, a large number of enhancer locus in human genome. So in 2 years ago, we actually performed the first pan-cancer analysis of enhancer RNA expression in TCGA patient. For that, we based on 15,000 enhancer locus identified by cage in Phantom. So I just brief set up, mention what we found in this paper. So using that enhancer activity, we can study the global pattern of enhancer expression, and then that help us understand some fundamental issues in cancer. So for example, in cancer, there's a long-standing observation that cancer actually can be classified into 2 general groups. One group, they really have a lot of mutation, but fewer copy number. They are driven by mutation, so they call it C class. There's another group of cancer, they actually have a lot of copy number, but they really have fewer mutation. So they are called C class, and then the group driven by mutation called M class. So this is very intriguing observation, but people never know what's the mechanism, at least the global mechanism underlying this kind of 2-class classification. So through our enhancer analysis, actually, we found chromatin state could largely affect this global pattern of somatic mutation. So for that nearly 9,000 tumor, we can classify them into a tumor with global enhancer activation. For them, they usually have this open chromatin, and then because this open chromatin, they have a lot of gene enhancer interaction. So as a result, they actually have this increased copy number. On the other side, when you have this they have active DNA repair, so they have fewer point mutation. in contrast, there's another group tumor generally don't have this global enhancer activation. They underline them, they have closed chromatin. So because their DNA are well ramped around the nucleosome, they have fewer DNA interaction. Therefore, they have fewer copy number change, but they don't have more mutation because the inaccessibility by the DNA repair machine. So using that enhancer, we also can build this enhancer gene regulatory network. So we start from this enhancer gene correlation. But when you observe this enhancer gene correlation, there are three possibilities. One is that the enhancer really caused the gene expression. That's called causal model. The other is it's that the gene expression affects the enhancer expression. That's called the reactive model. The third one is the enhancer and the gene, they show expression because they are a co-regulation, because they are co-regulated by a third factor. We call that co-responsive model. So to distinguish these three models, there's one way to focus on this EQTL signal, because only in that causal model, one SNP in the enhancer can show the EQTL signal in for the gene. So based on this principle, we build this model where we start from co-expression pattern between enhancer and the gene across multiple cancer types. And then we can use 1000 GTAX integrated EQTL signal to really tease out that identify those causal enhancer gene regulation. And then we also further incorporate high-seq data to identify those direct causal enhancer regulations where the enhancer location is close to the target gene. So based on that, we can build a gene enhancer regulatory network. So in this figure, one green box is the enhancer region. One gene circle is driver gene. So one important prediction we found is there's an enhancer for PD-L1. So as you know, PD-L1 is a very important therapeutic target for immunotherapy. So we try to understand whether validate this observation. So we could introduce CRISPR, double CRISPR knockout, so we can delete that enhancer 9 we infer from the genome. And indeed, you see in the qPCR, after you delete that enhancer 9, you see a substantial reduce for PD-L1. But then in Western blot, you see the same thing, even with the interferon gamma, you still see a substantial reduce the PD-L1. And then based on that, we can further propose a NF-kappa B mediated enhancer promoter interaction model. So where the NF-kappa B form a dimer, they bring this enhancer 9 close to the PD-L1 promoter, therefore turn on this region. So this is our first work where we focus on regular enhancer RNA. We just use phantom cage-based annotation for enhancer. But then there's a problem when we came to the super enhancer region. Because you know, when Richard Liang first published that, introduced the concept super enhancer region, now has been recognized super enhancer region play important role in disease development, include many cancer types. So basically, super enhancer region is where you have certain region, you have quite a high density of enhancer elements. So if you look at this CHIP6 signal, you see you run the enhancer region, there are certain regions where you have substantial, very extremely high density of CHIP6, indicate that they are very high density enhancer. So for such region, it becomes problematic how you quantify enhancer activity, because when you use quantify the enhancer region, because this region usually super enhancer region is very long, it could be several kb, even 500,000. For such a big region, you just cannot directly quantify enhancer RNA because the background noise will substantially confound the analysis. So one key thing is we want to identify the very clear unit within the super enhancer region. And then based on that enhancer unit, we can really quantify the enhancer activity. So for that, we perform a study. Here, just a general view. We focus on some core super enhancer region, which is about 377 megabase. And then we're using TCGA to study their region to try to understand the enhanced ERA expression pattern within the super enhancer region. From there, we hopefully form some principle. And then we can generalize this principle to a global identification to identify super enhancer locus. Here we end up identify 300,000 super enhancer locus. And then from there, we also try to understand how that enhancer locus are defined. And then with this high resolution map, we can carry out a series of applications. Okay, so for this, as mentioned, we focus on core set of super enhancer sets. They identified in at least 20 tissues. So we should mention there's a lot of super enhancer report in literature, but there's really a lot of noise. But really, when you focus on core set of super enhancer region identified in 20 region, in 20 tissue, they are really high confidence super enhancer. And then for each of that tissue, we generate a super deep resolution, super deep RNA-seq. So for each RNA-seq, for each tissue cancer, we aggregate hundreds of RNA-seq data. So you can imagine then for each region, you have extremely high coverage. Then we can focus on those super enhanced region. I just give you, so for example, here is one representative super enhanced region. And then here we just get one small piece of that several hundred BP. And then given this region, what you look at is we found there's so here each box represents one cancer type. And then for example, the first one is KRSE, that's a kidney. And then this one is KCM, that's a skin melanoma. And then what we observe, there's a recurrent ERA expression peak in the super enhancer region. And then that enhancer peak actually, they show very different cancer specific pattern. For example, you see this orange peak, they are very high in kidney, in breast cancer, in liver cancer, but essentially very low in this leukemia. So that's actually show very specific cancer pattern. And then when you look at all nearly 30 cancer type, you can see identify 5 different peak regions. So there are some peak like red peak, orange peak, green peak. They are all turn out in different cancer type. And then if you look at those peak regions, whether they are conserved in the other context, we can do the similar analysis in another large scale normal tissue, GTAX, where you compare their location, you found they are actually very conserved. Even in this 6th region, here just show the normal tissue versus the cancer. So the ERA locus, actually, they are very conserved, only have very little bit shift. So if you look at, if you get all those enhancer peak, you superimpose them together, you found those enhancer peak show the length is only about 100 BP. So they show very sharp reduce from when you walk away from the central region. So similarly, when you look at the GTAX, you also observe they have only the really length of this ERA signal only about 100 BP. So in this core super enhancer region, we actually can identify 30,000 enhancer peak. And then we try to understand, if you look at this, the TF signal in the super enhancer, in this ERA signal here, but show you the enhancer peak. And then we show the motif around this ERA. You see there's some very clear strong tandem transcription factor binding motif on both sides of the ERA. And then we also want to rule out the possibility that small signal just due to some unnoted exon, maybe that will due to splicing. But actually, if you look at the flanking region in the peak, there's no splicing motif. So we're pretty sure this is due to the, looks like a enhancer, this is enhancer. But then we also want to see how those enhancers are regulated. So we collect the nucleosome profile across 29 tissue on cell type. And then you superimpose the locus and the nucleosome position. So basically, you can think this y-axis is normalized the nucleosome occupation. So we should mention that RNA is very tissue specific. So when you have this enhancer signal, they usually only activate in one or few tissue. They silent in most tissue. So when you superimpose that RNA peak with the nucleosome signal, you see there's exactly nucleosome occupation corresponding to that ER signal. This is because in most tissue in that ER is silent, so you see occupation. So from there, we can think ER expression is regulated by a well-positioned nucleosome. So we further can look at those well-positioned nucleosome across different tissue, whether they are conserved in evolution. So there's some mouse and pig, they also generate this nucleosome position in multiple tissue. So when you look at the conservation of those tissue, you see those position, well-positioned, they are highly conserved in evolution. You generally can see when you have this nucleosome position, you see the peak. So that means even though this nucleosome enhancer signal, they are not conserved in sequence, their underlying controlled position, they are actually highly conserved. So based on that, we can have this kind of model where you have a well-conserved position in underlying each enhancer region. So when this nucleosome occupant, the enhancer are silent. But when this nucleosome off, the enhancer get this nucleosome-free region, then they recruit polymerase and then generate active transcription, generate the ER signal. So, so the difference between that nucleosome is if you have nearby nucleosome, but that position is more fuzzy, so that's called fuzzy nucleosome. So the difference for enhancer is really they have underlying well-controlled, well-positioned nucleosome. So based on that, we observe some, then we try to extend this to the global region, to the whole genome have nearly 372 megabase super enhancer region. We try to apply this principle. For that, I just want to remind you from firstly, number one, we see enhancer have this very short but sharp ERA peak. Second one, through our analysis, we found this underlying well-positioned nucleosome. So based on that two features, we actually performed the PCA analysis on nucleosome position on nearly 4,000 ERA peak in super enhancer region. And then they have three major components, P1, P2, and P3. One important thing is we found that there's a negative value of P3. That PC3 component, negative PC3, actually means a strong nucleosome occupation in that ERA peak. So based on that ERA peak and based on the nucleosome signal, here we use just PC3, we could identify nearly 300,000 ERA in 373 super enhancer region. So for each that super enhancer region, there's only about 120 base pair. So by doing that, now you can imagine we have very clear unit of that ERA unit. Each is very short, so we can quantify the ERA signal. So then we can further validate this super enhancer region ERS unit when you by cage signal. So basically, we can superimpose this ERN signal, and indeed, for those overlapped with cage, you can see a clear ER peak on both sides of the region. So that actually indicates, because the cage signal is totally independent from our ER identification, that actually validates we indeed have good signal for ERA. But I just want to show you that when you overlap our annotated ERA locus with phantom, only a 2 or 3% overlapped, and then the vast majority of them actually very different from very far away, like even 5 kb for those. There's more than 60% 5 kb from those known phantom. So the enhancer region we identified, they are very far from those. The vast majority, they are not annotated by the phantom, because phantom using cage, they only can annotate a small portion of the enhancer. But for those overlapped, we did see a good signal. So this from that mention indicate our enhancer map actually reliable. Then the key question is why we want to focus on enhancer. Because actually, Eric just mentioned that a lot of situations when we study cancer, there's an issue called tumor heterogeneity. Because when you really study the bulk sample, you have what you get is an average across different tissue. And then, but we believe that because enhancer is really confer that cell type specificity, so you can imagine that enhancer RNA provide a natural solution to address those heterogeneity. So here, I just give you a conceptual. So suppose you have a mixture of 3 types of cancer. You have cell type A, cell type B, you have enhancer A, enhancer B, enhancer C, where enhancer A in cell type A control GX, enhancer B, they have correspondence. And then what we really do in many cases is genotype phenotype correlation. Suppose there's a phenotype really controlled by cell type A. But when you look at the signal cross different tissue, when you really do the gene expression phenotype correlation, you often see a very weak correlation because the gene expression signal is average across 3 different tissues. But when you look at the enhancer level, because in the enhancer level, you have this, you already tease out this effect. So when you really see the enhancer A, you see for strong enhancer RNA signal, you have strong signal with the phenotype. So that's actually one way, in addition to single cell sequencing, which is not available in many cases. When you study the enhancer RNA, is strong complementary effort to identify to those signals identified in the coding region. So so here I can give you one example. For example, in cancer, we usually try to recently immunotherapy, especially PD-1, PD-L1, anti-PD-1 therapy has been a major driving treatment. But then there's a lot of effort to try to predict which patient will benefit from PD-1 therapy. So usually, for example, in melanoma, there's only 40% of patient response to anti-PD-1 therapy. So early stage people try to understand this feature. They generate the RNA-seq data from responding patient and non-responding patient, try to see if there's a signal, some gene expression signature can help predict anti-PD-1. Here, just show you on the left side, you have this response patient and the non-response patient, and then you could do a differential analysis. Here, just show you the p-value distribution. You pretty see, based on messenger expression, you see pretty flat, meaning there's really no significant messenger signal. Indeed, given FDR in the original paper, Cell paper, they did not report any significant gene. But the other side, if you based on our enhancer map, where you can quantify enhancer RNA in melanoma, you see a significant peak where you can identify hundreds of very significant enhancer show differentiation. And then this, if you base on those enhancer RNA signature, you can classify. You see, generally for those patients have complete response, they show much higher enhancer signal. And then in the non-response, they show no enhancer activity in the middle. So this super enhancer locus, based on that, you can differentiate this phenotype. And then you also, based on their related gene, you found their identified gene site enrichment. You found for those patients who have this response, they have increased expanding CD8 T cell, but they have decreased exalted CD8 T cell. So this is exactly because the immune cell is really depend on the T cell status. So in that case, the reason you see the response and non-response is in the tumor, the T cell they show the really play the determinant role. But when you really focus on the gene level, the T cell signal just dilute when you use the box RNA-seq data. But when you use the enhancer signal, because that enhancer locus, they are specifically expressed in T cell, you have much higher cell type specificity to capture that signal. That's why you can get the T cell signal. And then based on that signal, the enhancer signal, we also using that signal to validate that in the other immunotherapy. So in the other immunotherapy, it's called anti-CTLA4, which is secondary, another immune checkpoint inhibitor. So after that, you can see using our enhancer RNA signal, we inferred from CT-PD-L1, you also can predict the immune server response. So again, when the T cell exalted is high, you have bad survival. When you have T cell exalted low, you have good survival. So I hope through this, I just want to mention that we performed this systematic study of ERA expression in super enhancer region. And we found that super enhancer region actually contained that discrete ERA locus featured by a very sharp ERA peak, only about 100, 120 base pair. So the expression of such enhancer peak is regulated by dynamic, well-positioned nucleosome. So when the nucleosome is off, the enhancer transcription activation, they can generate enhancer signal, ERA signal. When the nucleosome occupied ERA locus, they turn off that. So this is like those well-positioned nucleosome is highly conserved in evolution. So we can, based on those principles, we identify high-resolution ERA locus through which super enhancer now can conveniently quantify by regular RNA-seq. So before our study, really there's no way to do the high resolution to the super enhancer region unless you really generate a super ChIP-seq data. You know, ChIP-seq is not as easy as regular RNA-seq. But now with RNA-seq, given this map, you can quantify ERA locus. And then we also argue that ERA signal can provide exponential power on quantitative traits beyond gene expression, especially when that gene signal really shows dramatic difference, variation across the different cell type. And then that ERA signal will, because they actually, ERA activity is the molecular basis for cell type specific gene expression. So ERA signal actually much easier to capture those power, exponential power. So with that, I want to just, so our paper just published in Cancer Cell. So if you want to know more detail, can read this paper. So I want to, basically, I just want to briefly mention our understanding is really through this pyramid. It's really when you have a better understanding about the genome structure. In this case, we have another set of annotation of super enhancer unit. We have better understanding about cancer biology, and then we have better understanding about the disease biology. Then we have clinical application. So through the example I show you today, I hope you like through this large scale data analysis, we first redefine the unit, basic functional unit in the non-coding region, super enhancer region. Once we have that, people can now use regular RNA-seq data to quantify super enhancer activity. And then from that, it's through comparison what happened in cancer versus normal tissue on different stage, on different response, you have better understanding about disease biology, such as in T cell, exhausted T cell, in no responder. And then from there, you can develop potential new biomarker to facilitate the selection of patients. So finally, I want to sum. So I have this brilliant postdoc Han Liang. He is really the hero for both this cell paper and the cancer cell paper. And then I also want to thank my other postdoc, Chun Yan and then Zhicheng, for experimental validation and funding resource. And then we'll stop here. Very happy to answer any questions.